Okay, so I want to start by thanking the organizers, particularly Nancy and Mario, for giving me the opportunity to speak. I think Jerry and I are very pleased to be representing some of the BLT humanized mouse work in the field. And I'm hoping to weave together a story for you of various experiments that we've done to show you some of the utility of the model, some of the things you can do with it, and hopefully something a little new. So just to orient you, I think this was already introduced by Rosie, but we're very interested in the concept of using vectors to deliver antibodies. So obviously, whereas conventional vaccines inject immunogens and rely on the immune system to make an immune response, here we're able to give adeno-associated virus vectors that encode monoclonal antibodies as an alternative to allowing the immune system to find that antibody. And this is done by encoding both heavy and light chains of an antibody within the confines of these inverted terminal repeats, which are 145 base pair elements from AAV2 that direct the packaging of this single-stranded piece of DNA into this capsid protein. And if you do this and you inject it into mice intramuscularly, from a single injection, you can achieve 100 to 1,000 micrograms per mil levels of these antibodies that are sustained for effectively the entire lifetime of the animal. And so this actually is now being tested in an NIH trial happening at the VRC called VRC-603, and hopefully there'll be information on that in the next year or so. But what we've been doing in the lab is modeling these interventions using humanized mice. And so just to get everyone on the same page with what humanized mice are, in the context of this talk, we're talking about the bone liver thymus mouse model, in which human CD34 stem cells are implanted into non-SCID gamma immunocompromised animals along with human tissues, including liver and thymus. And after this surgical implantation, you get a pretty extensive engraftment of human lymphocytes throughout the mouse tissues of all different hematopoietic lineages. And if you infect these mice with HIV, what you get are pretty standard things like CD4 depletion, and importantly for this talk, sustained viral load, which enables you to do things like look at the effect of antibodies on this viral load as therapy. But just to remind everyone that you can also use this model for prevention, I wanted to dig up some old data in which we used the BLT mice to show protection in a repetitive low-dose mucosal challenge study. So this was borrowing from the NHP field, this idea that you could repetitively challenge mice or animals and look at the number of challenges as your signal. And so here we prepare the BLT mice and then give them an AAV making a VRC07 G54W antibody, and then begin challenging several weeks later weekly with REJO.c, which is one of these transient founder stocks of HIV. And then ultimately harvesting tissues and looking in tissues and in blood over time at what happens, right? So in the serum using an AAV, within 4 weeks, you can get 100 micrograms per mil of this antibody. And importantly, you can do sampling of the vaginal site. So this is by vaginal wash. You can actually detect VRC07 G54W there. You can see pathogenesis of the virus here now at the end. When you sacrifice the mice and you look in their guts, you can see that they're lacking CD4 cells as well as in the vaginal lamina propria, so appropriate places that you might expect pathogenesis. And then ultimately, you can track over time viral loads to look at protection. So in our case, all of the mice in red here that were given VRC07, G54W, were unable to be infected despite repetitive challenges for 20 weeks in a row, whereas the controls became infected within about 7 or 8. This is just a more sensitive metric, so we really couldn't detect anything, even using clinical viral loads. But you will note that our limit of detection in mice is somewhat higher than what you're used to seeing in monkeys, and that's because, unlike monkeys, obviously we can sample only small volumes. I will say 50 microliters is definitely doable for those of you who are wondering. We actually sample closer to 100 microliters a week. And then this really leads to the main focus of my talk, which is what about therapy? So this is nice for prevention, but can you use the model in a different way? So here I want to show you a model of vectored immunotherapy in which we're using AAV to deliver antibodies to mice that are already infected. And the antibody that I want to start out with is one called VRC07, which may be hard for you guys to see in the back. but is a very potent CD4 binding site antibody. The basic setup of the experiment is now just reversed. So now we infect first and then give JR-CSF. So this antibody against this particular challenge stock is quite potent. It takes about 20 nanograms per mil to inhibit 50% NUC in the TZMBL assay. You can express high levels again, as usual, using the vector system. So as a control, we gave animals either luciferase expressing vectors or AAV JR-CSF. And so one of the great benefits of the model, obviously, is our ability to do extremely high-end experiments. This is a compilation of actually 5 different experiments that I've put together. But just to show you that the model does not lose infection, so you can actually infect mice, and keep them infected for up to about 6 months. But in contrast, if you give these mice an AAV that expresses JR-CSF, you actually see a pretty interesting outcome. And this is just a single antibody, right, which is unexpected, I would say. And that is that consistently there's a decline in viral load followed by 2 different outcomes, either escape and high viremia or low sustained viral loads for the duration of the experiment. And so we thought that this suggested that this was going to work extremely well. So we started testing other antibodies, including PGDM1400, which binds to a glycan and V2. And most people would say that this is a more potent antibody on average. It's not quite as broad, but binds a different spot. In our case, it's actually reasonably potent, almost exactly as potent as JR-CSF. But we could express fairly high amounts of it as well. But interestingly, when we use it in our study, none of the mice did what happened in JR-CSF's case, right? So all of these guys remained infected throughout the experiment. So then we thought we'd try more potent and broader antibody called N6. N6 is another CD4 binding site antibody, so it's like JR-CSF, but it's supposed to be a much better version of that. However, in our hands, it's actually about equally potent as JR-CSF against this REJO.c transmitted founder strain that we used. We can still express high levels of it, not quite as high as some of these others, but still many, many fold above the IC50. However, when we actually put this into mice, you see that actually none of the mice stay suppressed. So escape seems to be different depending on what antibody you use. And so just to summarize this data, essentially, of all the antibodies we tested, the only one that seemed to be able to actually suppress was JR-CSF. And if we look at the viral loads that were present in animals after we gave the intervention, you can see that there are clear differences. Now, to summarize a huge amount of work that went into repeating all of these studies using JR-CSF, I'll just tell you that nothing worked to suppress JR-CSF. And this was apparent even at the viral load levels, right? So despite the fact that we're using the same antibody that looks really good here, it doesn't work. So what is it that makes those 2 outcomes different? And that's really the subject of the remainder of my time in this talk. So just the key points so far are that you can actually give an AAV to prevent transmission, and you can use humanized mice for prevention studies, that you can actually suppress a subset of BLT-infected REJO.c mice with JR-CSF, and that equally potent or even broader antibodies like PGDM and N6 were not able to suppress REJO.c, and none of these antibodies that we tested could suppress JR-CSF. So what is it that makes JR-CSF uniquely potent? Well, another thing that you can do, just like in monkeys, right, is extract viral RNA and do sequencing. And so this is a representative example of some sequences that we got out of mice that had been infected with REJO.c and then treated with these various conditions. And I'm just showing you the OMS aligned here. And first thing you'll notice is that the luciferase-treated mice do pick up mutations. So this is actually a transfection stock. And we don't see these mutations in the stock. We definitely know that they're arising in the context of the BLT model. And importantly, we see new mutations arise when we treat with different antibodies, right? So in the case of JR-CSF, we actually see these 2 mutations. For PGDM, this is the one that seems to pop up, and for N6, that's the one. And we can re-engineer those back into the challenge stock and actually confirm their potency, right? So I'm not going to get into the details here because I have an easier way to summarize this, but ultimately what I'm trying to tell you here is that we've confirmed that these mutations do in fact lead to escape of these infectious molecular clones from the antibody that selected them. If you do the same thing with JR-CSF, you see a somewhat different pattern in that it seems like BR-CSF now takes only one mutation instead of two, but other antibodies all seem to only take one or two mutations to get away. Again, we actually re-engineer these back into JR-CSF and confirm that, in fact, in this case, it only takes one mutation to get away from pretty much everything. And then the next thing that we thought we would do to try and understand if there was a rationale for why certain mutations happen in certain contexts was to actually test the fitness of all of these viruses. So this is just P24 assays being done, growth assays being done in activated human PBMCs with the various mutants. And I don't want you to try and understand this slide. Essentially, what we're doing here is just measuring the slopes of these lines and using those as a measure of the fitness of the virus. And then I'm plotting those on what we're calling an escapability map in this slide. And so on the x-axis, I'm going to plot the resistance benefit of specific mutation. And on the y-axis, I'm going to plot the cost that we measured by those slopes. So just to orient you on the graph, a virus would start out down here. It would be both fit and sensitive to the antibody that we're going to test. If there's a mutation that actually makes it unfit but remains sensitive, it's going to end up here. Any mutations that grant resistance but are also unfit will end up in that corner. And ultimately, if you're both fit and resistant, you end up here in the lower right-hand corner. And this is where you want to be if you're a virus. So to map the PGDM escape mutations that we observed for REJO.c, what we can see is that REJO.c itself starts out down here. So this is normalized fitness to 1 and fairly high sensitivity to the PGDM antibody. And both individual mutations that we observed have fairly low costs here, right? So they can grow just as well as the wild type, but they grant full resistance from a single base pair change that makes a glycan deletion. N6 actually is quite interesting in that it actually starts out here and ends up in a single base pair change being able to go to A281D. And this mutation has a fairly high fitness cost. And what we observed, though, is that over time, we can find viruses that have adopted either a second base pair mutation at a different position or 3 completely unique base pair changes in one codon to change an alanine to a lysine and restore fitness. And ultimately, many of our viruses at the long-term late time points in our studies end up down here, as you might expect. And so what's interesting about BR-C07, which was, again, the only one that we were able to actually successfully use in our animals, was that these viruses seem to have to go through a somewhat intermediate stage of resistance. And then ultimately a second mutation that grants either higher resistance with not a lot of extra cost or greater resistance still, but at greater cost. And this was quite unique to the BR-C07 map that we didn't observe in any other context. And just to sort of show you what it looks like for JR-CSF, what we saw were, again, single base pair changes now, leading to complete resistance from BR-C07 with fairly minimal, in some cases, fitness costs. And likewise for the glycan antibodies like PGDM. Interestingly, N6 actually seemed to require 2 different mutations to escape, but there seemed to be a reasonably low fitness cost way to get there. And presumably over time, this would find further mutations. And then PGT121, another glycan-directed antibody, very easy escape, very low cost, essentially. So the key points to take home here are that escape seems to follow this multi-step evolutionary path for REJO.c and BR-C07 that seems to be associated with this ever higher cost of fitness, whereas in the case of JR-CSF, which escapes very easily, it happens in a single step, and there's pretty much no cost to making those changes. So this leads us to a hypothesis that effectiveness in vivo is not being predicted accurately by the in vitro potency and breadth measurements that people are typically using to score an antibody, right? These global panels and these breadth potency curves, that doesn't necessarily tell you how hard or easy it is for the epitope that antibody binds for the virus to actually make a change there. And that perhaps that might be a more accurate way or an additional way that we should be quantifying the potency of these antibodies in this concept that we're sort of calling escapability. So just in the last few slides, I want to go over whether this actually means anything in the context of a heart-suppressed infection, and hopefully show you that the model can also be used there. As actually Jerry showed as well, you can infect these mice with a virus and then give them ART and then apply your intervention and see what happens. So this would be a model of functional cure using an AAV. So we're able to, again, express high levels of these antibodies as usual. But in this context, very interestingly, if you treat REJO-C infected mice with JR-CSF, you can actually maintain complete suppression. But interestingly, if you use PGDM1400, which I already showed you is really easy to escape from for this virus, despite the fact that they were suppressed, and they actually are delayed relative to our control, which receives an irrelevant malaria antibody expression vector, you actually get fairly consistent rebound. But interestingly, N6 shows you a much better outcome than it did in vivo, during therapy. So in a therapy setting, you get rapid escape. It seems that perhaps that higher fitness cost of the first mutation to escape from N6 might be enough to actually keep most of these animals suppressed. In the context of JR-CSF, we can also show you that this somewhat recapitulates what we've seen before. So now JR-CSF is not effective. Remember, it takes a single mutation now to get away from JR-CSF in this context. PGDM, somewhat effective, but mostly not effective, and N6, completely ineffective. So we're essentially mirroring what we've seen in the escapability maps, even in the context of this sort of functional cure approach. And so just to conclude with this giant wall of text, I hope I've shown you that BLT humanized mice are useful for prophylaxis, to emphasize that they can actually be infected with authentic HIV strains, and they're all in the context of authentic human receptors. So one of the things Lewis said was that monkeys are more closely related than mice. I think that's not really the right argument, because here we have human cells inside of mice, and those obviously are human. And so all the interactions are happening between all human antibodies and human receptors. Ultimately, that you can support different routes of challenge, and that because of their fairly low cost, you can actually do fairly large N experiments and really precisely quantitate things like, for example, minimum protective doses that would be very expensive to do in monkey studies. You can use these mice to do therapeutic interventions, and you get sustained viremia for up to 6 months, and fairly high viremia that enables you to do things like sequencing over time, weekly sampling. You can actually obviously harvest them for tissues. So you can do things like look at evolutionary paths. In the end, importantly, it's fairly absent of endogenous immune responses. So this actually is a bug in the model, I would say, the lack of humoral immunity. But in our case, you could consider it a feature if you want to study how antibodies actually escape in the absence of other selection pressure. This is a model in which you can do it. BLT mice also let you do things like test cure strategies that target reservoirs, because you can use standard HART regimens to suppress them, and they actually get quite consistent rebound, and obviously you can apply your intervention. And then just to sort of emphasize some of the downsides, so as I mentioned before, this bug/feature is a bug in the context of vaccine testing. So if you want to actually test your favorite vaccine, I would not start out in the BLT models currently, although I would say that's an area that there is room for improvement, and there are ways to improve these models to improve immune function, and we think hopefully become a reasonable place to test vaccines. They tend to have low innate immune cell reconstitution, although this is graft dependent, and again can potentially be enhanced using other interventions. They do have GVHD, which can limit the very long-term experiments, but there is a window of time before they have GVHD where you can actually do some good science. And I think the biggest downside to me right now is this new restriction that we have for fetal tissue, which has created a real cloud of uncertainty for anyone who wants to use this model and is hanging their career on it. No one quite knows right now what's going to happen when they send a grant in and it gets reviewed by an ethics panel that no one really knows who's going to be on and how they're going to make decisions. So anyway, with that, I just want to thank all the people who did all the work, and particularly mention this team of technicians, Christine, Evan, Dylan, and Scott, who did almost all of the sampling of the mice that you saw there. And this work was really led by Kaylin Deal, who recently left the postdoc in my lab. So I'm looking for postdocs. If any of you have great grad students that you want to send my way, I'd be happy to take them. And then hopefully there's a next meeting, and next time the meeting picture can look a little more like this instead of like this. And with that, if there's any time, I'm happy to take questions.
So Alex, one of the things that expensive monkey studies and very expensive clinical studies have shown is that it may be important to use 2 antibodies at once. What do you think about that, and can you do that in this system?
Yeah, definitely. So we've been testing combinations in the system. I think that's one of the big advantages is we can iterate and test probably more combinations than are reasonable to test in larger animals. And so we could be a useful place in which to figure out something that looks promising before moving it on to an NHP study.
Hi, Jonas Asher from OHSU. Nice talk.
Thanks.
Did you screen for anti-drug antibodies or ADAs? Because as is shown in macaque studies, that is a huge problem that has since been confirmed in humans with the PG9 factored AAV, where people made anti-drug antibodies in circulation.
Yeah, I'm glad you asked, because I get this a lot from people who work on monkeys because they read the monkey literature, and if you've read a lot of Ron DeRocher's papers, you would think that this approach is dead in the water, right? Because when you try to put human antibodies into the monkeys, or even immunized antibodies in the monkeys, they're a disaster. In the mice, you don't really have that problem. The vectors actually seem to generate tolerance to the transgene in some ways. So AAV8 is known to be somewhat tolerogenic in a mouse, but not in a monkey. I would note that the PG9 study and some of the early things that Ron was showing were done in AAV1 capsid, not AAV8. And AAV1 capsids also are not quite the same in terms of their induction of tolerance. So some of the more recent things that Mario and Rosie were showing with Hugh was that if you used fully simian antibodies that were derived from monkeys and deliver them with an AAV8, you could limit the ADA, I would say. I mean, it definitely looks very different than Ron's studies. It's not 100% perfect, but it's certainly much better. So the current clinical trial is using AAV8, which I hope, will have a different outcome, but we'll find out.